Today we're going to talk about meiosis, and the goal of this video is to describe the process of meiosis. I'm going to give you an overview, and you're going to learn how to predict the consequences of crossing over and explain how this introduces variation into the gametes that are formed. So first, what is meiosis? Meiosis is a special type of cell division that produces gametes. Gametes are sex cells. And those sex cells are used in sexual reproduction. In humans, the sex cells, the gametes, are sperm and eggs. Sperm is the male gamete, and eggs are the female gamete. Now, you should note that gametes have chromosomes in them that are made up of DNA, and DNA replication happens before meiosis does. Now, why is that important? That's important because that means that any mutations in the DNA can end up in the gametes that are produced during meiosis. Okay, I've said this word meiosis a few times, and you might recall that previously we've discussed mitosis, so I thought it'd be helpful to lay out some of the differences between mitosis and meiosis. Mitosis makes genetically identical cells, but meiosis makes genetically different cells. In mitosis, cells go through just one division. So you can see we have one cell here. It's going to divide, so at the end you have two cells. But in meiosis, cells go through two divisions. So you begin with one cell, it's going to divide, you get two. Then each one of those divides. So by the end of meiosis, you actually get four cells. So last difference here, I said that mitosis ends with two cells. It's important to know those two cells, they're diploid. But meiosis, the four cells that it ends with, they are haploid. So what's the difference between diploid and haploid? Diploid cells have two sets of chromosomes in them, but haploid cells only have one. So that's the significance of the 2N and the N that you see. Wherever you see 2N, that means there's two sets of chromosomes there. So this cell would be diploid, this cell would be diploid. But the cells that you see just an N written in, they only have one set of chromosomes in them. So very, very briefly, I'm going to go over the steps of meiosis with you. You don't need to know all the specific details of what's happening in this step, but I want you to at least see in pictures what's happening during meiosis, because that's going to help us later to understand where the variation comes from that you get in the gametes. So meiosis is going to have a total of 8 steps, and I've listed those on the left here. You may notice they repeat, so it goes prophase, metaphase, anaphase, telophase, and then it repeats again, prophase, metaphase, anaphase, telophase. Remember, I told you meiosis, the cell divides twice. So this is the first division, wherever you see the number 1 here. And then down here where you see the number 2, that's the second division. Okay, so I'm going to quickly go through each step and describe what's happening. So in prophase 1, the nucleus breaks down, chromosomes form, they condense, and homologous chromosomes pair up. So, for example, you can kind of see this is a pair, red and blue, that have paired up, and then over here there's a pair that have paired up. And when they pair up in prophase 1, crossing over occurs. So I've used a couple words here, homologous chromosomes and crossing over, so let me tell you what those mean. Homologous chromosomes are chromosomes that are similar in size, shape, and genetic content. So you can see these are roughly the same size, this red and blue, and these are roughly the same size and shape, red and blue. So that's why they're a homologous pair. And crossing over is where those homologous pairs of chromosomes, they exchange genetic material. And when they do that, this creates a new combination of alleles that are on that chromosome. And that results in variation. So up here, in this top right corner, I'm going to show you a crossing over event. So here we formed a homologous pair. Notice that the ends are going to overlap and they'll actually swap places. They exchange material. And so now you have new alleles on this chromosome and you have some new alleles on this chromosome. That's going to lead to variation. That's crossing over. Okay, so the next step of meiosis is called metaphase 1. And this is where each pair of homologous chromosomes lines up on the equator. So here we have a pair, here we have a pair. They're lined up right down the middle of the cell. The spindle fibers attach to those chromosomes. And then in anaphase 1, that pair of homologous chromosomes get separated from each other and they move to opposite poles, opposite ends of the cell. In telophase 1, the spindle fibers, those strings, break down and a new nuclear membrane forms. That's what you're seeing in yellow here. And then the cytoplasm here in the middle is going to divide. So now we don't have just one cell anymore. Now we have two. So you see a cell on the left and a cell on the right. And here begins the next cell division. So each one of these is going to divide. So now we're in prophase 2. The nuclear envelope starts to break down. That's why you see this dashed line, dashed circle. That nucleus is breaking down. The spindle fibers are reforming. That's the things that look like strings. And the centrioles, these things that the strings are attached to, they start to separate. In metaphase 2, the spindle fibers pull the chromatids to where they're lined up right in the middle along the equator of the cell. Now, I used a word there, chromatids. So you'll notice here's a chromosome, but the left side represents one sister chromatid and the right side represents one sister chromatid. So in metaphase 2, the chromosomes are lined up down the middle of the cell, and each chromosome is made up of two sister chromatids. In anaphase 2, those chromatids get separated and they go to opposite ends of the cell, opposite poles. And then at the end here in telophase 2, a new nuclear membrane forms around those chromatids, and the cytoplasm divides. So you can see the cytoplasm dividing here, the cytoplasm dividing here, and that's going to generate 4 cells total. And each haploid cell has a unique combination of genetic material in it. And you can see that. So this one's mostly blue, but it has a little bit of red. This one's mostly red, but it has a little bit of blue. So these are different from each other. So to finish up, the process of meiosis in male organs is going to be a little bit different than the process of meiosis in female organs. So, and we name these processes different names to distinguish the two. So spermatogenesis is what occurs in the male organs to produce sperm. So this happens specifically in the testicles, and all 4 sperm are functional. For females, oogenesis occurs. This produces 4 cells, but one of them is called the ovum. That's this one. The other 3 are called polar bodies. Oogenesis happens in the ovary, and the ovum is the only functional gamete. So that gives you an overview of the process of meiosis.